Thanks. Now it's recording. Okay. But merA, then it's believed to be evolved like long time ago. But in the contrary, merB is believed to be recruited relatively recently. And probably it didn't originate for hydrothermal vents or like these extreme environments. So maybe the origin it's hypothesized to be more mesophil environments, and because a lot of bacteria until now it's believed to be absent of these merB genes. So usually in the operon you find merA but not merB, but maybe this trend is now changing a little bit because of the new findings that we have. Usually merB it's present in habitats or environments where there is a higher concentration of methylmercury, and also it has been seen that there is a uh, I don't know how to say it. It's not a correlation, but environments where there are mercury methylations, there are also like the ones that they have HGCAB genes. We also found merA and merB. So it's like a detoxification system for bacteria to cope with the methylmercury that other bacteria are forming. Um, Now we cannot here. Okay, sorry. So this is just a huge table that you can find in this nice paper from Christakis that it was published in some years ago. But it's like a recopilation of all merA and merB genes that we have in databases. And if you see that table, you can see that there is a bunch of taxa that codifies for this merA and merB. And if we look into the phylogenies of these merA and merB and the genomes that goes for these genes, not all of them code for both genes. Some of them only code for merA, and some of them only code for merB. So, as far as I know, it is said that usually genomes, there is a higher ratio of genomes that code for merA than for merB, as you can see also in this Venn diagram. But new findings in different environments, especially, for example, sediments, it's seen that this ratio is a little bit different. So we find more merB than merA. So maybe our identification, that it's the thing that we are here to discuss, it's a little bit biased because of the environments that we have sampled so far or we have studied so far. And maybe if we move to different environments, this ratio changes. Also, the taxa that we found there, it's different. And also then the merA and merB that we found there are a little bit different. And we have to take this into consideration if we want to identify them properly. So, if you look into all of this, then different questions arise. It is necessary to have both genes for activity or for the demethylation, for the correct demethylation? Are alternative reduction pathways if you don't have the merA, or alternative demethylation pathways if you don't have the merB? But maybe this is something that we can discuss maybe in another session. But something to pop up, or if you want to discuss it, it's okay for me. There are some also in this paper from Christakis, he also summarized a little bit some alternative mechanisms for reduction of the mercury too, in case that bacteria doesn't have merA. I thought that this table was quite interesting because if you, for example, in the case that in sediments we found more bacteria with merB but not merA, what is the mechanism that they have to cope with the mercury too then once they demethylated? But then to the thing that we are going to discuss more in detail, it's how can we detect those genes? So mainly, I just put this two main techniques. So mainly we have PCR or qPCR that it has been used in a bunch of studies. So you use primers to detect the genes that you are interested in this case, merA or merA and merB. And also you have metagenomic and metatranscriptomics. So with metagenomics and metatranscriptomics, you sequence all the DNA that you have or all the RNA that you have in a sample, and then you specifically search for the genes of interest, in this case, merA and merB. There are different advantages and disadvantages of these two techniques. So maybe I can share you also later these slides, even if we are recording, but maybe I can share you if you want, because I will not go in detail with all of this, but mainly with PCR and qPCR, we are a little bit biased because it's primer dependent, so we need to really take into consideration which primers we are selecting, and sometimes we will not get all the diversity depending on the primers that we are going to use. But also with metagenomics and metatranscriptomics, even though maybe we detect higher diversity, it's a little bit more complicated, in terms of analysis, data analysis later. So I think it depends on the scientific question you have. Maybe you can use one or the other and choose. So, but in this case, we are going to focus on the metagenomics and metatranscriptomics part. So I have tried to summarize and in the bibliography all the literature that I could find, different methodologies that have been used until now to detect merA and merB. And mainly all the methodologies that we found, they are based on the construction of hidden Markov models and the verification of the merA and merB genes by looking to essential amino acids. So hidden Markov models, for those who are not familiar with this term, is like a statistical model that allows you to identify specific patterns in DNA or RNA. But it needs to have like reference database. So you are also biased with a little bit with the database that you are choosing and the reference sequences that you are choosing. And then the other step, once you have identified the merA and merB, it's to correctly identify those that are functional. And according to the literature and different papers, there are different essential amino acids that are necessary for the correct functioning of the merA and the merB. Here I put the positions of these essential amino acids for merA and merB. For merB, there are some that are optional or they are thought only to be structural. So also it depends how strict you are with selecting those essential amino acids. Maybe you lose some of the sequences that you detect. And also these positions are also based on reference sequences. So these are the most common used reference sequences for the verification of merA and merB. But maybe if you use another one, you have to take into consideration that this could change a little bit. But these are the most common ones. So there are also a bunch of papers that have used these hidden Markov models for the identification of merA and merB in different environments. There are not a lot, I must say, but I think we are going to see more and more coming, also focusing on different environments. So now I thought to put here some of the available tools that we have and that we as a community could use because they are or public available, or maybe we can ask our colleagues to have access to these tools. So for example, this pipeline was developed by Eric Capo. And it was intended for the detection of HECAB genes, but also he optimized it also to detect merA and merB genes. So you can find it in his GitHub, and it's public because they publish also the paper. I forgot to put here the reference of the paper, but it's very straightforward pipeline that you can use to detect merA and merB genes from raw data, or both from metagenomics and metatranscriptomics. And you can have like an output table where you will have the abundance of the different merA and merB sequences that you have. And also you can have a taxonomic classification based on reference trees. So I think it's a great tool to use. And as far as I understand, I think they are optimizing a little bit this pipeline to be really focused on merA and merB, but I'm not really sure. And then, okay, so this pipeline will be more for the detection of the merA and merB, but as we said, we also need some verification because sometimes we will detect sequences that are not true merA or merB. Because one thing I forgot to mention is that the diversity of merA and merB is so high that it's difficult to detect them. So even between um, different strains from the same genus, sometimes they have different merA and merB. So it's quite difficult to detect them. That's why we do these hidden Markov models. So we need to do this verification process. And then one of the last papers I was working when I was in Barcelona was this, where we detected merA and merB genes from the bathypelagic deep ocean. And we published this mer verifier script. It's a Python script mainly. It's very easy to use, and it's public, so you can use it. And it's based on reference sequences for merA and merB. And then you can just select which essential amino acids do you want to choose for the verification. And you can also change the reference sequence if you want. You just need to upload the FASTA of the reference sequence. So if I don't know if it's now because we wanted to update its README file, so it's more straightforward to use for users that haven't used it before. But if you have any question, you can contact me, or also I think Pablo, who is the one that uploaded, he will be very happy to help. But yeah. And then I know that there are other people working in verification methodologies or pipelines or scripts. I think Nicola from the University of Porto, he's also working. It's as far as I understand, it's very similar to the mer verifier script that we have. Maybe it's a little bit different because they built a score table based on the amino acid position of the sequences that you have. But it's also based on these essential amino acids. I think that they will publish soon some results based on these methodologies. So, or maybe you can ask them if you need to use it. I don't know if Nicola is here, but maybe he can say something later if he's here. And then I think it was in the last seminar session that we have also Zohra, she shared with us the methodology that she was using to identify also merA and merB. So for example, in this case for merA, she was using this motif that it's a metal binding motif, and she found that it was quite good to identify or verify merA genes. Um, And then for merAB, she was looking specifically for these two systems that it's also included in the other verification tools that I told you before. But then she went a little bit a step forward, and then saw that always there is like a 63 different like separation position between these two systems if this is not the correct position in the sequence. Unfortunately, I think she's not here because she couldn't join the seminar, but maybe we can talk with her a little bit more in another occasion if this is interesting. Um, because I believe she did some manual inspection of the sequences also in order to check this, and I think it's a good work. And then, so this is the slides that I had prepared so far, with the methodologies that we have now. So one of the main questions that I had when I was using these methodologies and also when I was preparing these slides is that maybe we are too strict with the verification process because we need to have all the essential amino acids to believe that these sequences are okay, because we don't want to have false positives, but maybe we are losing some that they are really functional, but we don't know because we cannot test it in the lab. Sometimes we will need the isolates, and we usually don't have access to all the isolates, of course. Also, another question is if our identification it's biased based on the databases we have. So, what I don't know. Maybe now we can open our mix and say what we think. If you have any input on this, any ideas.
Or also if anyone has like any questions before like
Yeah,
that
true.
about what Isa has presented. Well, I can start. I yeah, that's fine. Uh, I do think that the databases that we use are well, making our findings completely biased. Um, I don't, I, I, I don't know as well how could we test in silico, like without having the actual isolates, if methylmercury was the substrate of the enzyme or not. Because I know that Tamar Barkay made some has some papers about the different types of enzymes, but these are quite old papers, and I don't know if they have the sequences like the amino acid sequences for those uh, different enzymes. I don't think so.
I
So
don't
I don't
know.
know how good we even test this.
Yeah, that's a good point. It's like as you said, it will be useful to have the isolates, but we don't have it for all of them. I don't know if there is any way to test it in silico, but it could be a good idea. But yeah,
So with the
I agree
transporters,
with
for
you.
example, there is like for the transporters, there is known that certain transporters only transport certain compounds. So merE, for example, only transports methylmercury. So maybe a way would be to check the sequence, like where merE is actually binding the methylmercury to transport it, and see if like different enzymes have that pattern, but not other enzymes have it, something like that. I don't know. The easiest thing would be just to test it on the lab, like find an isolate that has tolerance just to methyl mercury and not to any other form of organic mercury, and just
Mhm.
like get that sequence.
Mm. But again, because merA and merB are so diverse between strains, it behaves different
Yes.
depending on the strains.
Yeah, and also like if I'm not remembering wrong the paper, most of the like isolate enzyme isotypes, or I don't know what's the name, but like the different types of enzymes, they work for like multiple substrates. It wasn't
Hmm.
just like one substrate in
Hmm.
general.
Hmm.
So yeah, that will probably won't work. I don't know.
Mm. Any other
Isa,
thought?
yes,
Yeah,
thank you, Isa. I have a question. Like, do we know, yeah, actually, what is the percentage of genes that we considered after the Maki Coco, for example, that we consider MER-A and baby, and they are not? I mean, what is the error that we are doing here after the Maki
yeah.
Coco?
It reduces a lot.
Okay.
Like I don't know the exact percentage, but um, it's at
It
the
also
end.
depends on your environment, I think.
Yeah.
Mm,
For example, for seawater samples, the ones that I'm more used to work, you could detect like thousands of sequences, and at the end, you like the true merA and merB, it's 100 sequence. Like it's reduced a lot.
mhm,
When I passed the Marquicoco for for the samples that Marta and I are working with now, that was just it was just like a preliminary check. So that would be need to like corroborate better, more efficiently. But just like open numbers, I had like 30,000 supposed Mer A's,
Yeah,
and none of them passed the verifier,
yeah,
like 0%.
yeah. That's something that's why maybe we are too strict. But then I also was working a little bit with manual verification just to see if we were missing something or some of the sequences were shorter, and that's why we do not have some of the amino acids in the extremes of the sequence. And for some of the sequence, it's the case that they are shorter, and that's why we don't detect all the essential amino acids. But for some of them not. So the hidden Markov model is detecting um, other sequences that are not MER-A and MER-B. Uh, but also because, for example, for MER-A, it's a flab independent. If I don't remember wrong, so it's very similar to other kind of enzymes that the bacteria already have, and that's why we are detecting them. But then it's
Maybe
not true.
another metal
Yeah,
enzyme, like.
exactly.
mm. Andrea said on the chat, there is very little known anaerobes described, so we are likely very strict for mere anaerobes. That's what we meant as well with the types of environments that we studied.
Mm,
So these essential amino acids were based on studies that were uh, mainly done in waters.
mm.
So yeah, probably we are considering that all of them need to be the same, like the same essential amino acids,
Yeah,
and that's probably not the truth.
yeah. That's why maybe when I was also preparing this presentation, it depends on the actual environment that you are working. Maybe you need to choose properly the reference sequence, like which are the organisms that you could trust to be in that environment and that have similar positions of the essential amino acids, because it's not the same sediments or soils than freshwater or marine waters.
Yeah. What database did you use when you did the identification?
Uniprot
Okay.
from
Could we filter like directly from Uniprot, just say everything that comes from soils or sediments, use it, and the
I
rest
think you can.
not?
I think when you download, you can know the origin. Most of the samples they say the origin. So I think that you could construct a database like reference sequence for maybe more specific for sediments. Of course, there are shared taxa, but yeah.
Yeah, the taxa is also not very useful in this specific case
Mm,
because they are horizontally transferred, so
mm,
taxa doesn't really say as much.
mm.
Yeah. It's but when the so when you did these like reference database, Marta Ferri did it, right?
Yeah.
Do you know if she actually checked that the proteins that she was using for the reference database to construct the hidden marker models were functional, or were just like what's available on Uniprot? Because a lot of the things that are available on Uniprot, they are, for example, if you do the annotation with Proca, sometimes Proca tells you like, okay, this is a merA, and then it doesn't pass the verifier. Like it doesn't pass the
I think
verification.
she checked.
Okay.
Yeah. Because she was dealing a lot with this database. So I think she did.
Okay.
Yeah. But even the Marky Coco, I think it uses a different database.
Probably. I think Markiko uses the Christakis database.
Yeah. So I think it's like for sure it's like a recent version, and as far as I understood, they are updating also. So it should be like a recent version of the database to deal with this.
The script that uses Marta Ferri, the verifier one,
Yeah.
don't use a database. What she did was taking a reference sequence and
Mm.
doing a multiple sequence alignment with your sequence that you want to check and focusing on analyzing each position.
Yeah, but
But
I think Carla was talking about the previous step, the
Exactly.
hidden.
okay, the previous step is for
That's the only the verification step. But first you need to do the hidden Malcolm models to actually get the query.
yeah, exactly.
Yeah.
Yeah.
But thanks, Esther.
Have you used it there in your samples?
Yeah, me, I recently could retrieve this information, the merAB data, and I was doing the ratio because I have the data done obtained for how many merA genes have been found with Coco and how many merB without passing with verifier, and the ratio is like 90 to 10, well, 90 to 10 merA merB obtained from raw Coco. But then after filtering, what I get is a 60 to 40. Like there is a strong filtering of merase when passing
So
the
there are
verifier.
more genes that pass the merB than merA.
Exactly.
That's what happened as well with the samples for my thesis.
But the thing is that my samples are mixed, are sediment, water. The most of them are sediment and biofilms. So, but I will have to make a classification in order to know in which cases have been filtered more merase, more berries. But yeah, in general, it's more sediments and biofilms.
Okay. I will just put the next slide because I think I had more open questions here, this one. So we can comment again in the things that we are saying, but just you have it here. But I was thinking that I don't know if someone is working on this now, but should we create a common methodology or at least a common database that we all use so we can also compare papers or results from different environments? I think at the beginning, Eric Capo wanted to do also a common database, like, but I think no one had time to work in that. So I don't know how is this. Um,
Is that what he is doing now? Do you know?
he is not doing, but maybe someone that he knows is doing. But I think he's not working on that specifically. He's more, as far as I know, he's more focused on the methylation part, not the demethylation part.
Okay.
To my knowledge, he's not doing, but I think he collected already a large amount of information, so because he really wanted to.
Mhm.
I think having the common database allows more people to study, and then it's easier to compare across studies and across systems, because then we want to know if we have really new organism or different diversity, or compare oxic waters, etc. It's easier. It also makes it more available to the mercury community, which, as you can see, we are not many people doing microbiology. So
Mhm. Yeah.
I think in the mercury community, this is still a gap.
A lot of the people that do mercury microbiology don't do mer. Like they focus more on the methylation side,
Mm.
right? Like, isn't that like general?
Yeah, I think if you look at just Google Scholar, there are more papers focusing on the methylation than the methylation. But I think it's important that we could also have this common database.
Yeah,
Mm.
I agree.
But most
Oh,
likely, one of the main reasons is, on one hand, uh, so historically, it was very difficult to detect the merA and merB genes because it's
Mm.
so diverse, right? That we are not completely sure if they are or they are not. Also, because at the beginning we were detecting them only in high contaminated sites. So now,
Mhm.
when we are studying more ecosystems that are not contaminated, we have been overlooking the merged genes. But of course, the paper of Bowman that showed already the presence of the merged gene in the ocean, and then with Isa's paper, we have clearly seen that they are more widespread. But still, we are all, or we are limited by the primers we use, or we are limited by the detection with the metagenomes, because we don't have a common database, and people working on mercury in general are not people doing microbiology and metagenomics. So it's because I have the feeling that we are opening this
Yeah.
now. So it
Mhm.
can be a nice moment. As of course, for the AGCA, it was very obvious, and of course, we were searching for this gene for so long that then it appeared in 2013, and we all started, okay, we need to do the primers, we need to search for the sequencing, and it opened a lot of new knowledge, of course. And then people focus a lot on that, and when you work on a data set, you can see that it's not easy to do both. At the end, we end up writing papers on AGCA or merA and merB. Some papers combine, and it is very nice, but it's not so easy, because then to find a message, it comes complex. So I think it can be really helpful. It was one of the ideas that with Eric we wanted to do, but of course it's a lot of time. It's engaging.
Yeah.
Mm.
Agree.
So I am part of the people that don't know so much about microbiology, but I'm still interested about it. And I'm gonna go to Canada and work with Alex Poulain. And the objective will be to look at this demethylation pathway in marine sediment. So I started some incubation or enrichment, and that will be very like focused on the like how it is linked to methane oxidation as well. So I mean, it's gonna be specific, but I hope that I will be able to bring some information about that. And I see now how this merging is not that easy to work with. So I don't know where I'm putting my feet, but I think it's interesting. It's challenging. I see here some research
She's thinking
person.
of switching, just staying in Stockholm instead.
Yeah, I like the laugh, like, but yeah, I think it's interesting. I do believe that we can probably. I don't know if I have the expertise or like I'm good enough in this field to help, but I hope that I would find some and give some good information.
But Alisa, yeah, after a few months with Alex, you are gonna be
Yeah,
able to do it, no problem.
yeah,
The idea with the Marquicoco at the end, first it was the Mercury Mata database, no? So the version 2.0 would be with the mergings.
Mhm.
But so at that moment, it was Benjamin, it was Kathleen, it was Eric. So it was really so many people are here also, no? That participated. So it was a completely bottom-up initiative. So they worked together and they combined all the knowledge they had to take the sequences from the isolates, from the from the marks, and build it. Um, I think it's a very nice thing that then you can ask for help. Some people can explain. They distributed the task. There is already some of this work has been done because Eric collected a lot of information on the merging. So perhaps it's worth to ask him.
Mm.
And then, for sure, he's not gonna lead it. He's overloaded of work, but perhaps he can actually help or give some hints of how to do, and then it's completely up to the community to build
Yeah.
it.
Yeah.
I think that would be good. I
Mm.
mean, it's good if, I mean, it's how research work, right? You start stuff and then you never, there's so many frustration, but this, if you can give over like some work and it can grow, it's still a nice thing.
Yeah.
It would be good. Okay.
I think that maybe we can send an email to the Mersortium community and see if there's people interested in trying to join and forces and then construct this database. And then once we have if we have a response of different people, maybe we can organize ourselves.
I like the idea.
Yeah, and that would also help with not being too strict. Like if the first step is to make the database, but if we have mercury genes from different environments with different gene lengths, and there's also a lot of isolates that have been tested, like tested to have available concentration tolerance and everything. There's a lot of work done with isolates in the mercury, in the merAB genes, much more than with AGCA, which is extremely hard to cultivate. But a lot of the things that tolerate mercury, like that have merA and merB, are pseudomonas or pteromonas or marinobacter, or that
Yeah.
grow like anything. You look at them and you can see them grow. Like it's ridiculous.
Yeah,
So
that with
there
this
is
I
also a
agree.
lot
I
of information
agree.
available on actual functional enzymes.
I agree with you in the side that we have really good studies with isolates, but then, as you know, isolates are a few percentage of the community. So I'm afraid that maybe we miss the ones that are not growing, and they are different because they have different metabolisms also. So, but yeah, we can try to think different
Mm.
options.
But just maybe a stupid question. I know that there is all these AI tools nowadays. Is it any way that it can be used to like facilitate the like filtering or like finding this like um, genes or like for creating this table. I'm just really like just in terms of creativity, how we could improve it, because it feels like we are always like having the same problem. Can we just find another path to make this table?
Probably yes, I don't know. I'm not an expert
I
on
don't
that.
know. Maybe Marta knows.
I
Do you
think
know?
that I think that in order to develop AI tools for this purpose, you should have a data set that has been trained that is more or less representative of different environments. So I think that the problem here is with the data that we are beginning with, like the small representation of reality that we have at the moment. We could train AI tools if we had more representation. If not, I think that we are just prolonging this bias,
Mm.
like shifting these predictions with a hard bias from starters.
Okay.
Isa,
And
I wanted to ask you a question. Oh, sorry.
oh no, go.
Yeah,
No,
yeah,
it's fine.
go. No, I was
It
gonna
was
shift up.
okay. I'll leave you ask.
No, Alisa, go ahead. I was shifting a topic. If you wanted to continue on that, we
No,
can.
I was just wondering, like, because you showed like the work from Zoa, I think it was her name.
Yeah.
And then you mentioned that it could be also an alternative. So what is different with her approach?
Um, maybe we can ask her in another.
Okay.
But I think that for merAB, it's quite similar what we were doing. Also, Nicole, I think it's quite similar for merAB identification. But for merA, she's not only identifying the essential amino acids, but she's identifying this metal binding site. So it's more just like a fragment that it's inside the sequence and not like essential amino acids that are uh, spread on the sequence. I don't know if I explain myself. So maybe we can have a combination of this metal binding motif and the essential amino acids. But as far as I understand, to avoid false positives, it will be more accurate to use the essential amino acids than to use this metal binding motif.
Oh,
But
okay.
I didn't work with this, so with this specific pattern, so I don't know if the results would be similar or not.
This motif also contains two of the essential amino acids. So the
Yeah.
two Cs could be part of the essential amino acids.
Yeah.
Okay. So please, Marta.
Isa, thank you. I was wondering, I'm super new to the Mercury world, and I will study Mer A and B. So this session was super cool to have just for like an introduction and getting right at the state of the art of the topic. And I was wondering, based on your expertise and knowledge, if there has any study that looks at the genetic context of these merA and B genes instead of just focusing on the sequence of these genes, since it's very, as I understand, like transferred easily horizontally. I was wondering if has people looked at the gene context of the cells that are actually functional at demethylating with these genes that we can use as markers maybe to be identified in other genomes that we cannot isolate and that we cannot test, and whose MER sequences may be a little different, but maybe the location is exactly the same, and that could also be a clue.
Yeah. I'm not sure about the answer, but I would say there are not a lot of studies. Like if there's something, maybe it's in isolates, as you said, but not. I would not.
There is
I
one
haven't
that is
seen.
in isolates. It's a paper from a woman named, well, last name Ibars from 2008. And she looked at the genetic islands in two species of Alteromonas. And she did look at the context, like genetic context of certain islands that did have the mercury genes. But I don't think it's very like representative of the general.
Because this paper that you are mentioning, I think she found it like because she was studying Alteromonas strains,
Yeah,
not because she was
yeah.
studying.
She wasn't studying
Mm,
mercury. Um, she was studying genomic islands actually. She
mm,
just like had a whole paper
mm,
studying, like looking at the differences in the genetic islands in two strains of Alteromonas.
no,
But
I
but
was,
med
yeah,
genes are horizontally transferred, so their genetic context, it's always different. No, I mean the
that's why
logic.
I mean, I meant like genetic context in a wider sense, like talking genome wide.
Genome wide, okay.
Yeah, like maybe just to understand other kinds of biases that we may have in our studies. Maybe these genetic islands are accepted and included in the genomes because they can be added to the genome, but the genetic characteristics or in terms of the composition of these genomic islands differ too much from the genomes which host the islands, that we may find these genes, but they may not be so easily transcribed because the cell's machinery does not work so well for these
mhm,
islands. And I don't know, I was thinking that maybe there's some kind of structural characteristics of the genomes that we work with that may be also altering gene expression.
yeah,
We somehow, I think, in Carla's paper, the isolates you work with had 4 different MER operons, and we could see that one of the operons was not working very well.
Mhm.
I don't know if this is because of the MER genes within the operon or because where the whole operon was inserted in the cell was perhaps
Mhm.
not suitable for making that operon functional. But that could be checked, for example. These are data that are available.
Mhm.
Yeah, this is available. Also, there is just the possibility of, we discussed it in the paper, that that operand didn't have merB, and our experiment was with methyl mercury. So one possibility is that it's not transcribed because it's not needed,
mhm,
Mhm.
because it doesn't have merB.
yeah.
So yeah.
But I think there's not a lot of people working on that because as far as I've seen in the Mercury community, we are very focused in the Mercury. I think so.
Because we already have hard time handling
Yeah.
one gene with our knowledge that of course we miss a lot of the genetic context of the organism, no? But we are still getting on that. Yeah.
Yeah. But
Marta,
it's a good point.
how would you approach that if you wanted to start doing that? How could you do that?
I mean, that's since I will be working with Max from Venice, that's one of the things that I wanted to check. Also asking, like, to see if there was something already done that it hadn't passed and I didn't know of. I don't know. I think that first, like checking basic characteristics of genomes like GC content can be one indicator of GC content is an indicator of like the kind of metabolisms in very general terms and the kind of environments that cell thrives in. Like number of like general characteristics of a genome in terms of sequence, of transporters, of location of these islands. They may be located next to highly transcribed regions of the genome. So by chance, those genomes just felt at the right place to be highly transcribed. I don't know. I think that also gene alignment, sequence alignment can be really helpful, but like it can be humanly approached in short sequences, like for example, these genomic islands. I don't know. I mean, I need to think about it, but I think that it could be interesting. Of course, we may get negative results, but we could also distinguish either biogeographic patterns or, I don't know, genome patterns that can be associated to certain types of merA and merB genes that may be functional or not, but maybe more indicative, for example, than doing a taxonomic placement with a reference alignment. With genes that are included in these genomic islands, I don't think it makes much sense to place them taxonomically, as you have already been talking. But maybe there are some structural patterns that we can find somehow.
Great.
That'll be really cool.
Yeah. I don't know if someone else, because we are almost approaching the end of the meeting, if we want to keep it 1 hour. So I don't know if someone else has something else that they want to share or comment or ask.
I can say that it was a very good session, and I really appreciate that you took the time putting everything together. It was really good.
Thanks.
So thank you.
Thanks.
And before leaving, yeah, I think it's really useful, and I think we really have to think, I mean, seriously to build this data set, because it will be really useful. It has been like the data set with a GCA-B is really, really useful, like almost every day for all of us. So I think it will be really cool to have something similar with the merAB in it.
Yeah, I think that we can send an email from the Mersortium community and see if people want to join to do this. And maybe we can meet another time and see. Yeah.
If so, I would, besides this, I would directly also contact Christakis, for example, Ben, Kathleen, etc., just in case they miss the email with the Mersortium. I think they might be really helpful. Don't know if they would like to be involved or not, but for sure they may be really helpful, guiding or sharing.
Good. So I think I will stop recording now.